Hello students, welcome back to a new video of Sunanda's tutorial. So in this video we will discuss about the transduction process in bacteria. So it is a mode of horizontal gene transfer. Okay, so what are the other methods of horizontal gene transfer in bacteria? So basically it is of three types. One is transformation, second one is conjugation, third one is transduction. So here in this video we will discuss only about transduction. So this transduction part is particularly present in your plus 3 first year major as well as minor syllabus of botany students. Okay. So it is a process by which a bacteriophage or bacterial virus transfer genetic material from one bacterium to another. Okay, so it is a special type of horizontal gene transfer, or otherwise known as parasexual reproduction, where the gene transfer takes place from one bacterium to another by a bacteriophage. A bacteriophage helps to transfer the DNA. Suppose this is one bacterium and this is another bacterium, and it is the bacteriophage. What is bacteriophage? Actually, bacteriophage, it is a virus, and the name is bacteriophage because they infect. They have the capability to infect the bacteria. They can infect the bacteria, they can kill the bacteria. Okay, so in this process these are the genes of bacteria. So they at first transfer their DNA into the inside the bacteria. Then they cleave that bacterial DNA into pieces. Finally, bacterial DNA convert into small fragments. Then after that, this bacteriophage create mini bacteriophage inside the body of the bacteria, and finally break down the cell wall and release into the environment. So the new bacteriophage formed inside this bacteria have bacteria DNA, have viral DNA. So when those infect another bacteria and again release the gene inside that bacterial body, they release this bacterial DNA to this bacteria. So in this way the gene transfer takes place. So at first this mechanism was discovered by Ladderberg and Ginder, Joshua Ladderberg and Norton Ginder, in the year 1952. Okay, so they discovered this process when they are doing their experiment with Salmonella bacteria. Okay, next, what are the steps? So at first, attachment. So attachment means the bacteriophage attach on the surface of bacteria. This is the bacteria, this is bacteriophage. They attach on the surface of bacteria. Then injection. In the process of injection, bacteriophage inject their DNA into host cell. That means inside the bacterial cell. Then break down enzyme from the bacteriophage, break down the host DNA. Okay. Next, replication. The bacteriophage replicates, creating new bacteriophages, and that may include some host DNA. So now the diagrammatic representation of the whole process. At first, the bacteriophage attach with the bacterial cell surface. Then they inject their DNA inside the bacteria, and then the whole bacterial DNA is cut into pieces. Then new bacteriophage formed. Some are with viral DNA, some are with bacterial DNA. Next, step 4, this bacterial DNA, the bacteriophage having bacterial DNA again infect another bacteria, inject that piece of DNA into their body, then that piece of DNA get integrated with that new bacterial DNA. So in this way the genetic recombination occur in bacteria by the process of transduction. Okay, it is a very special type of pathway. Okay. And here another thing, we have also studied in my channel about lytic and lysogenic cycle. Okay, so it is a very important process where lytic cycle is involved. So here lytic cycle is involved because new bacteriophages are formed, and they lyse the cell and finally release into the environment. Okay, so in this way one lytic cycle is involved in this process. Okay, next. So there are two types of transduction. One is generalized one, where the bacteriophage infect the bacterial cell, inject their genetic material inside the bacterial body, bacterial cell. Then they cleave. The enzymes help to cleave the bacterial DNA into fragments. Then the new bacteriophages are formed. Some are with viral DNA, some are with bacterial DNA. So in this way they finally cleave or break the bacterial cell and release into the environment. The next one is specialized transduction, where the bacterial cell, some bacteriophage transfer a specific set of genes from one bacterium to another. That is the case of specialized transduction, where the bacteriophage transfers a specific part of gene of one bacterium to another. Okay, next. Actually, in generalized transduction, normally the bacterial cell receive the bacteriophage DNA, then finally help to cleave the bacterial DNA. Some bacteriophage formed with viral DNA, some with bacterial DNA, and they finally release into the environment and infect another bacteria, release that DNA, bacterial DNA of this bacteria into that bacteria, and so in that way the gene transfer occur. But in some cases, in case of special type of transduction, either a specific part of gene gets transferred, or in other way. At first bacteriophage release their DNA, then lysogeny takes place. In the process of lysogeny, the viral DNA gets integrated with bacterial DNA, and that integrated part then finally inserted inside new bacteriophage, those which are formed inside the bacteria, and they finally release into the environment. So in this way hybrid DNAs are formed and gets inserted within the new bacteriophage. Okay, so this is the special case of transduction. Okay, so it is all about transduction, bacterial transduction. It is a very special type of method which involve the bacteriophage during the gene transfer. Okay, I hope you enjoyed this session and will get benefit in the future. And if you like it, please hit the like button, please share with your other friends, and subscribe to my channel Sunanda's Tutorial. Thank you.